So I am a graduate student at the College of Engineering here in Iowa, and transitioning a little bit from cytotoxins, I'm here to talk a bit about some pharmaceutical exposures in effluent dominated streams. So first, I just want to orient you guys and remind you that wastewater treatment plants are not perfect. They do really, really well at getting rid of those nutrients and those bacteria, but they don't do so well getting rid of those emerging contaminants like medications or detergents or pesticides. So when that happens, they go through the wastewater treatment plant and then are just released in the environment pretty much in their native or in metabolite format. And that's a big concern because these effluent dominated streams can cause de facto wastewater reuse, which simply put is just accidental recycling of the water. So if the same stream is being used for source water for a drinking water plant as a wastewater treatment plant put their waste into, it's possible for those downstream customers to be drinking that contaminated water. And it's actually really common in the United States. It was first thought that this was only happening in the arid Southwest, but as you can see, it's really common in these temperate areas. Looking to Iowa and to Illinois, it's pretty common and happening in a couple cases greater than 15% of the time. Now, focusing just on pharmaceuticals, each individual pharmaceutical that gets put into the environment from these wastewater plants can undergo attenuation. And then as a whole, all the pharmaceuticals together undergo differential attenuation, where each pharmaceutical is not breaking down at necessarily the same rate. And some of these attenuation processes can include sorption with the bed sediments, or have some groundwater recharge and return, or even through photodegradation just from the sun. And then we also have processes like bioaccumulation, where the pharmaceuticals might be going into the bodies of fish or spiders or any other aquatic biota in the stream. And then we also have the process of biodegradation, where bacteria can be breaking down these pharmaceuticals. So moving on from here, I'm going to talk a little bit about someone's from our lab, her PhD research, and then at the end kind of get into where I'm going to be going with it from there. So overall, she was looking for what is the differential attenuation of these pharmaceuticals in an effluent dominated stream, and then how are they affecting these aquatic biota through these evolving exposure mixtures. So this was a really collaborative project started back in 2017. We had collaborators at the USGS in both Iowa and West Virginia. And then, so our contacts there were Dana Kolpin and Luke. And we also had a contact at UW Milwaukee, Dr. Rebecca Clapper. She did a lot of the fish studies for us. But today I'm just going to focus on these chemical analyses because that's what happened in our lab. So first I will orient you guys to the study site that was chosen. Muddy Creek is a really low order effluent dominated stream near Iowa City. The effluent is coming from the North Liberty wastewater treatment plant, where it's actually a really cool plant because it was only built in 2004. So it's relatively new compared to some of the infrastructure around the United States. And it also has this really cool state-of-the-art membrane bioreactor facility. So even with this membrane bioreactor, that means it's really, really good at getting rid of phosphorus and nitrates and bacteria. But even then, it's not so good at getting rid of those pharmaceuticals. Once the study site was established, they then established 4 sampling sites. So our first one here is an upstream site. So this is not affected by any wastewater. And then right at the effluent discharge, samples were taken. And then again at the downstream one. All of these are about 100 meters apart from each other, and then followed by this downstream 2 site, which was located about 5 kilometers downstream at a USGS stream gauge. And it was really important to have this USGS stream gauge during the whole process because samples were only taken during base flow conditions. And the reason that these base flow conditions were targeted was because these wastewater effluent dominated streams, especially in Muddy Creek, can be very flashy. So during periods of high rainfall, there tends to be a dilution of the actual wastewater, which means there's a dilution of the pharmaceuticals that we're looking for. So during base flow conditions, it's like the worst case scenario for the environment, but the best case scenario for us as scientists to figure out which pharmaceuticals are present. Now, moving on a bit to analytical methods, which I don't have to spend too much time on because Stuart so nicely explained all of this triple quad to us. Starting with the USGS, they took monthly samples starting in September of 2017. And what they did was target 109 different pharmaceuticals. And the reason that they targeted this many is just because the Denver lab has a method set up for the 109. But for us, we don't have the same chemical standards. So what we did is focused on 14 pharmaceuticals. And the way that they came up with these was through, sorry, it's really windy out there. So these target 14 samples came from, they were the most abundant and found in nearly every single sample. So it was easy to only look for 14 rather than looking for the 109 and still get really strong results. And then what the PhD student did was she went out with an undergrad helper, took samples every other week for 2 years, processed them with solid phase extraction, and then ran them on LCMS. Now, some of the results were really cool that they got from these only 14 pharmaceuticals. Each bar here that you can see is representative of the pharmaceutical mixture as a whole. So this is a total concentration, not individual compounds. And if you look at the blue area here, this is indicating the cool weather conditions. It almost looks like the concentrations are higher than in these warm weather conditions as indicated by the orange. But what they found out after normalizing to the flow conditions, well, that actually wasn't true. The chemical concentrations were actually greater during the warm season in the summer than the cool season. And that was mostly due to the use of antihistamines, which are seen here in the purple. And another really cool thing that they found was during the warm season, attenuation, how fast the chemicals go away, it was actually more rapid during the warm season than it was during the cool season. So even though there was more chemical concentration at that time, it went away faster. Another really interesting thing to look at is just the composition of the chemicals in total. As you can see here in the pink, that is the antihistamine. And as you flow away from the effluent site, so even just 100 meters downstream, a lot of the antihistamines have attenuated and gone away, but many of the antidepressants still persist. And this trend is seen for every single sample site, which is really interesting. Now, that is also seen in this here to explain some of the complex exposure mixtures. So the differential attenuation, that's coming from, if we look at just metformin and citalopram here. So metformin is an anti-diabetic, citalopram is an antidepressant. Right at the effluent site, they're both pretty abundant. Metformin is the top 2 and citalopram is the top 5. But as you go to the downstream too, so this is 5 kilometers downstream, the concentrations decrease, especially for citalopram, but not as much for metformin. That one didn't attenuate very much at all, really. That can be also seen in this graph here. Looking at the blue, this is right near the wastewater effluent, and the red is at the gauge 5 kilometers downstream. So you can see that citalopram really reduced its concentration a lot over that 5 kilometers, but for metformin, you can't say the same thing. Now, that research was really cool, but I am more interested myself in looking at some of the effects of a couple of the other things that they were looking at the same time. So during this collaborative effort, the USGS and Milwaukee had some fish studies going on, and one of the fish studies include this electrofishing, where, as you can see here, one of the native fish was captured from the stream. So any fish that was not considered endangered was collected and put on ice for later extraction to figure out what is the actual body burden of pharmaceuticals in their bodies. So that is one of the things that I'm working on for my own research. So I'm really interested to know how many pharmaceuticals are actually going into the bodies of these fish and what's the best way to figure that out. So I'm currently working on an extraction method to figure out their concentrations. But if anyone has, if anyone's done this without using SPE and they have really good recoveries without a lot of lipids left over, you should send me a message so we can kind of figure that out. I'm also interested in just figuring out what is in the creek as a whole. So we know that the animals near the creek are going to have some body burden of these pharmaceuticals. And we also know that there's medications in the creek and that there's pesticides in the creek. But I'm interested to know what else is possibly in the creek too. And I'm going to do that by using that non-target mass spectrometry that Stuart alluded to earlier. So I'm hoping that by using that, I'll be able to put together the puzzle pieces of maybe some novel metabolites, maybe some new chemical features, and just try and figure out what is happening with the creek as a whole. And then if this works well for an effluent dominated stream, I'd also like to try it in streams of other land cover types as well. With that, I'd like to say thank you and just give a shout out to Hui. This is her PhD research as talked about before. And then Jack and Grant, they're both undergrads helping with sampling. And then my PI, Greg, who's also very supportive. Thank you.
Thanks. We have about 4 minutes for questions.
Okay.
Okay.
So can you conclusively state that people use more antidepressants during the cool season, or is it possible that there is a differential behavior of other pharmaceuticals during that time?
Yeah, there could definitely be other behavior happening. I believe there are some papers out there saying, when medications are used. That's not specifically my research, so I can't say with for sure. Sorry, I couldn't think of the word there.
I was wondering if you've looked at impacts of those chemicals on fish species yet. I couldn't remember exactly where you had said where you were at with your studies.
Yeah. So most of the fish studies were done by the USGS and Milwaukee, and they're actually working on those now. The last I heard is that there's some preliminary studies saying that there's biomarkers in the fish that can be used to figure out if their estrogenicity levels are being impacted. And they actually found out that the estrogenicity levels in the upstream fish were more impacted than the estrogen levels in the fish downstream of the effluent, which is exactly opposite of what they were expecting. So that's one of the big reasons why I'm going to be doing that non-target analysis, just to figure out what other chemicals are in the creek that's causing those weird effects.
Okay, we have another question.
Mm-hmm.
Has the ingestion of human birth control affected fish reproduction?
I don't know about reproduction. Sometimes the fecundity of the fish is impacted. There's definitely papers out there focusing only on the estradiols and like 17 alpha estradiol. However, for this study, we found that it wasn't one of the top 14 compounds. So we can't say conclusively that they were affecting the fish in Muddy Creek, but in other creeks, there's definitely an impact of those birth controls.
Can you comment on what you might discover with non-target versus targeted MS methods?
Yeah. So for targeted methods, we're only specifically looking for a list. So like in this case, we had that exact list of 14 compounds. We're not going to find any more than that. We're only going to find those 14. But with non-target, you open up the possibilities to all the libraries or to just feature matching. And although I might not necessarily be finding exact chemicals, I might be finding co-occurring classes. So depending on if the metabolite or the chemical has been found before and put into a library, I might be able to say with pretty good certainty that something like PFAS exists in the creek. But then there could also be something like an obscure metabolite of benzotriazole that no one's ever studied before. And I might be able to say with lower certainty that something like that does exist in the creek too.